Okay, welcome everyone. Today, this is our third seminar talk for our Junctions of Statistics and Biology series. Today, we're going to have our speaker, who is also my student Miles, who is going to talk about another important computational task regarding single-cell RNA sequencing data. So, which is the data you have heard of in the first two talks. Okay, so Miles, maybe you can start from here.
Okay, so hello everyone. Thanks Jessica for this opportunity that I can give you this presentation about my current research. So my name is Nan. You can also call me Miles. I'm a fifth year PhD student in UCL Department of Statistics. I'm working with Professor Jessica on computational genomics, especially on single-cell RNA sequencing data. Uh, so here today, I want to introduce the topic, which is benchmarking computational doublet detection methods for single-cell RNA sequencing data. So I think in this topic, there's two things we may be interested in. First one is what is computational doublet detection? Okay, so that's the method itself. And the second one is how we can evaluate all the current methods. So the second point is probably relatively new to you guys because this is not to develop a new method, but to evaluate the current method. The reason we want to do this kind of research is that in computational genomics fields, there are a lot of new methods coming up in recent years. Uh, and all those methods, the authors of those methods are coming from different backgrounds. Some of them are from statistics, some of them are from computer science, and some of them are coming from biology. So they just use different methodology, is which is good for their own field to solve the same problem. But and also in their papers, they try to claim their advantages. But because there are so many methods, and there's a lot of difference in terms of those methods design and their actually performance in the downstream analysis. So here we want to do a comprehensive benchmark to compare all those methods so that we can give our users a guidance which kind of method is better and which kind of method is more appropriate for their goal. So that's the second point of this research. The first one is method itself. The second one is to evaluate all those methods. Okay, so let's move on to the second page. Okay, this is the outline of our talk. First, I will introduce the background of single-cell RNA sequence and the doublet formation. So you may already see this figure before. This is a illustrative figure to show the pipeline of doing single-cell RNA sequencing experiment and how to generate the data from the experiment. Uh, let's just briefly go through this. So basically, you're starting from the sample or tissue, and then you want to get some cells from those tissues, right? So you do some dissociation to get those single cells, and then you try to isolate all those single cells so that they are separate, and then you let those single cells go through some machines, and then you label each cell so that you can know what are those cells, and then you get all those RNA from those cells and get sequencing. And finally, you will get a data matrix. And in this single cell isolation step, we will have this doublet formation problem, which is you actually capture two cells or even more cells in one single volume so that you thought this should be one cell, but actually you capture two cells. Then you know your data would be wrong, right? You have two cells and you think this is one cell, so you actually create a fake cell. So that's the problem. And there are some experimental methods try to solve it, but they are expensive, and you have to take more time to get those special design. And so people try to use computational method, just generate the data and try to identify within this already existed matrix, which of them are doublets, right? There are fake single cells. There's just 2 cells or 3 cells. So this is the goal of doublet detection. And also that's why we have this problem. So here is try to further illustrate how we form doublets in this single cell sequencing pipeline. So first, in order to catch each single cell, you will have this equipment, which you have 2 flows. The first one is a horizontal flow. All those yellow beams, they are just the reaction volume. This is the thing you want to capture the cells. And in the vertical direction, you actually have cells, okay? And these cells will go up, down, and then those volume will go from left to right. And then when they intersect here, those volume will capture all those cells, right? So they will go through this pipeline, and at the end of the day, you will have those volume, and within each volume, you have 1 cell plus something else, which is we call it microparticle. So those yellow balls is to give you the labels to label all those cells, right? You want to paste a label, say, to 1 person to show, oh, this is who, this is your friend, this is your parents. So this is same as here. You label each cell, right, with a UMI code. So which cell will have a unique name. And then after you do the sequencing, you know, okay, this kind of RNA slice or RNA information is coming from this cell, and another piece of RNA is coming from another cell. Okay, so then in this interaction or intersection procedure, we will have this doublet formation problem. If these 2 flows are too fast, then probably sometimes there's 1 reaction volume here, and there's 2 cells come here together, then you will catch 2 cells, like those kind of circles, right? So there's a volume and there's 2 cells inside. Okay, so then you will get many of them as single cell, but some of them as doublets, so 2 cells in a volume. Then when you do sequencing, you still label those 2 cells as 1 cell, right? Then after you obtain the RNA, you treat those doublets as singlets. So then there's some problem because it will bias your downstream analysis. For example, in this figure, we actually want to perform a clustering analysis, which is you compress the data from high dimension to, say, 2 dimension, right? And then there's a property you want, which is similar cell will come closer to each other, and the very different cell will set apart, so that same cell, similar cell will give you a cluster, like this cluster or this cluster, right? And the cells between these 2 clusters, they are very different, right? So, and then people usually think that for 1 cluster, because they are similar, so they probably coming from 1 cell type, right? Blood cell or bone cell, muscle cell. Then sometimes people find, oh, these clusters, all those cells are very different from previous known cell types. Then people will say, okay, we find a new cell type. So that's very important discovery and very valuable property of this experiment to find a new cell type. However, if you have doublets, then it usually happens that all those doublets will give you a cluster, because all those doublets are very different from, say, single cell, right? Because there are 2 cells. So you will have say this cluster in the middle, they are actually doublets or they are fake single cells. However, if you ignore this problem, you will think, okay, this is a cluster, right? And if we compare all those single cells, fake single cell inside this cluster, oops, you will think this is a new cell type, but actually they are not. They are just experimental errors. So that will give you a problem. However, if you can identify those doublets and remove them, then you will not have this problem, right? Then you can trust your clustering. All those clusters are cell type, different cell types. Another example is trajectory analysis, right? So sometimes we know cell is differentiating in some process, so there's continuous change, say along a timeline. So if you try to project cell into low dimension, you will see there's a trajectory like this U-shaped, C-shaped trajectory here. So all the cells are continuous changing. But if you have, say, doublets, then you will find some doublets very different from those trajectory cells, right? So they probably appear in the middle of these two branches, and we call these bridges, right? Because they connect these two branches. And you will say, okay, there's a main trajectory here, but also there's a smaller trajectory in the middle, but actually the middle one is totally wrong, right? There's no such cell existed. You just randomly give those doublets and give you a new cell, but those cells are totally didn't exist in humans or animals, right? So that's all the problem that doublets can give you, and our goal is to remove all those doublets. Okay, so people propose a lot of computational methods try to find all those doublets and remove them. Here, there's a list that show you all those current methods. Uh, uh, you see here, they are very new, right? So most of them are within 2 years. And they are probably, most of them are published on top journals. So people are really care about this problem. And our goal is trying to benchmark all of those methods, try to give users a comprehensive understanding which method is better and which method is more appropriate for their applications. Okay, so we will benchmark all the methods except the last one, because the last one is just appear online and without publication. And also we already finished our paper. So there's no last method, but we will cover first 9 methods. Okay. So here I summarize all those methods in this table. We will not cover the very detail, but here the message is we have a lot of methods. And if you check their platform, you will find that most of them are developed under Python or R. So which means, so first, these 2 languages are very popular in data analysis and also in bioinformatics. And then another message is all those methods use different methodology to implement their algorithm. So that's very different methods. And here I just briefly summarize all those methods. I categorize them into 5 categories. The first one, I call it a KN-based method, which KN means K nearest neighborhood method. So the idea is actually very intuitive. You have the data, right? And then you try to see, okay, I want to find doublets, and I know doublets is just the 2 cells together, right? There's nothing special. So then how about I simulate those doublets, right? I randomly pick up 2 cells from my data set and combine them together. So I have doublets, even though there's not real doublets, but it's very close because we know doublets are just the 2 cells together. So then this method just randomly add 2 cells, 2 single cells. Then you get doublets, right? Then the next step is I want to see among all my current cells, which cells are similar, are most similar to these simulated doublets. Okay, so if you find those most similar cells, then we have high confidence that those similar cells should be doublets. This is our logic, right? So then, for example, in this figure, the red one is synthetic doublets, and then you still do some dimension reduction and project your cell into this two-dimension figure, right? You have X and Y, and it will give you a property that similar cells or data points will come together, come closer. So you will see, okay, some red doublets are closer to those kind of cells, but for other cells, they are far from those red cells, right? Then we will know, okay, probably those say blue points or cells, they are because they are closer to those doublets, simulated doublets, then probably they are more likely to be doublets than say other cells far from those red points, right? And to quantify those say similarity, we can just give a distance. Say you just draw a circle, right? If you are the cell and you draw a circle and you count how many simulated doublets within that distance, and you calculate the rate, right? Say 80% of my all my neighbors are actually those simulated doublets. Then you say this 80% is your doublet score, right? Then say if you only have say 1%, then if you compare these two scenarios, you will know, okay, 80% for you, if you have 80%, then you will be much more like doublets, right? Because similar people come together, so you are doublets, okay? That's the first category. And the second category is here, the second figure. I call it classification-based method, okay? Still, the idea is we want doublets, then we simulate doublets. So the first step is same. You just randomly pick two cells and add them together, then you have simulated doublets, right? And then the next step is different. Then you have true label, right? You assume you have true label. You have a single cell and the doublets. Then you can do classification, right? You can use machine learning algorithm, try to differentiate between these two classes, right? You can train a classifier, then this classifier can predict, okay, which given each cell, how possible that cell belongs to, say, single cell classes or doublet cell classes. Of course, that doublet is coded, right? Because you define, you simulate. Then you can just train, after you train these models, you apply those models on your data set and try to see, okay, because those models can give you the probability being one category, right? Then you can know, okay, this cell have say 80% probability to be classified as singlets, and that one will have 10% as singlets. Then the second one, it will have 90% as doublets, right? So the second one is more like a doublet. So that's the second kind of methods. And then we have the third one, which is statistical test-based method. I will not cover the detail, but the idea is you apply some statistical test and use the p-value as the score, okay? Sometimes if you have high p-value, then you just classify, you think, oh, this is more like a doublet, okay? And then the fourth one is deconvolution-based method. So this one, I also will not cover the detail. Basically, the idea is you just separate for each cell is a vector, right? High dimension vector. You separate them, make them into say several sub-vectors and try to see which one is similar to your simulated doublets. Okay. And the last one is ensemble-based method, which is there's no new method here. Just you combine existing method to give you a better method, because people believe if you combine two relative good method, you can get a very good method, which will surpass the previous two. Okay, that's the idea. So that's the general categories of current method. Then we will go through our first benchmark, which is overall detection accuracy on real data. Okay, so then you will ask, what is the real data? Real data is just a single cell RNA-seq data, but with experimentally validate doublet labels. People have some technique to use experiment to label doublets. Okay, so we have labels. So here we use 16 data sets, and you see here they have different data size and different gene numbers, and also different number of doublets. Okay, so we try to use all, this is the current available data set with doublet labels. Okay, we try to include all of them so that they are diverse enough to give you a more comprehensive picture. Okay, then we will apply all those 9 methods on these 16 real data and try to use them to identify the doublets, right? And we compare the identified doublets with the true doublets. And then naturally, the better method will give us more consistent, right? To be consistent with the true doublets. So we have 2 figures here. The figure on the left, we have 2 subfigure A and B. So basically, it is a box plot. So each box plot is a distribution of your matrix on 16 real data set. Okay, so we use 2 matrix. First one is AUPRC, and the second one is AURC. So we don't need to know the detail, but basically it tells you the overall performance of your detection accuracy on your real data. Okay, so the higher the better, and it's between 0 and 1. If you have 1, you have 100%. Then you have, if you have 0, you just do nothing. Okay, so and the x-axis is the different method. And also we include 2 baseline methods, right? So baseline is the most naive method. So there's no computational design. It's just you count how large the cell is. If it's larger, it is doublets. That's your logic, okay? So basically, because the box plot will give you the general distribution of all those 16 performance. So then you can very easily to see here we have the doublet funder and doublet detection and the solo relative better than others. Okay, and the second figure is we go further. On y-axis, we list each of those 16 data set, and we use the color to show the performance. If you have darker color, then which means the metric here is higher. If you have a lighter color, then it's worse. Okay, so you can see here, not only you can see some method is better than others, say still doublet funder, but you can also see the performance on different data sets are also very, very different, because the first several data sets they are very light, right? So which means the performance is not very good. But on the bottom one, you can see they are darker. So, okay. So basically, from this figure, you can very easily to see how the method performed. And then on the right one, this is a different perspective, because on the left, we just compare the general performance, but also people want to see here we ignore the absolute number of doublets you define. Okay, but on C we actually assume we detect some number doublets, and to compare those doublets with the true doublets, try to see the difference. So here we assume all the methods will identify 10% of all those cells as doublets, and 20% and 40% in terms of doublet score. If you have high doublet score and it's top 10%, then you just classify them as doublets. So and here we compare precision, recall, and the true negative rate. So precision just means among your detected doublets, what's the proportion of them are true? right? And recall is among all the cells, among all the cells, among all the doublets, how many you can detect. And the true negative rate is among all the singlets, how many of them you can correctly detect. Okay, so anyway, so for all those metrics, the higher the better. Okay, and also you can see here, basically we have consistent performance. The one on the right, doublet funders and solo doublet detection, they are performed relatively well. Okay, so here is the general performance. There's one method, doublet-decon. This one, it cannot give you doublet score. It just give you the identify the doublets. So to compare this one with others, we use this doublet-decon to decide how many doublets in each data set. And we let other methods to choose the same number of doublets and to compare their precision recall and the true negative rate. So in this case, doublet-decon will have some advantage because you let it decide how many doublets you want to detect. However, compared with others, this method perform relatively poorly, and still we have doublet funder and solo relatively well in this comparison. Okay, so previously we generally measure the performance of those methods, and the next one, we want to see under different experimental settings how they're performed. So before we just use we call it static comparison, right? You just have 16 data sets, and you just compare. But then this part, we are more like a dynamic comparison, right? We change one dimension of your experimental setting, try to see how the difference, how the trend of each method. So if you check this figure, it's very clear. Here we compare two perspectives of your experimental design. The first one is doublet rate, how many doublets you have in your data. And the second one is the sequencing depth, which means how the resolution of your sequencing. In other words, if you want to use image, is how clear your image is. And then how many cell types in your data set. And then heterogeneity means how different between those cell types. Okay, we compare different methods based on these 4 dimensions. Let's take the first one. We increase the doublet rate from 2% to 40%, which means there's more and more doublets in your data set. And we compare the performance of each method. And we actually find that with higher doublets, all the methods generally perform better than low doublet rates. Okay, and still the better performer is those several methods doublet funder, so which is consistent with previously. And then if you check sequencing depth, still the trend is going up, right? If you have more accurate measurement of your sequencing, your method will perform better. And for cell type, so still it's going up, but it's not that significant because so generally speaking, if you have more cell types, then the doublets will be easier to find because people usually think the doublets, if the 2 cells coming from 2 cell types, then it's easier to find because this is very different from singlets. However, if you have smaller number of cell types, most of them will coming from one cell type, then it's not easy to find. And heterogeneity is still, it is similar to cell types. If your cell type are very different, then the doublets from those 2 cell types will be very different from your single cell, right? So if you have high heterogeneity, then your performance will going up. So that's what we find here. And then we want to check the next important point, which is how they impact your downstream analysis. Because finally, you want to apply the method on your data set, and that's not over because you want to continue. After you clean all those doublets, you want to do downstream analysis. Then we want to check how your doublet detection can improve your downstream analysis, which is biased by doublets. Okay, so this is a general idea of how we can improve clustering. So first, this left figure show you if you have doublets, what will happen, right? So say we really only have 4 cell types, those gray clusters, and all those right points are doublets. Okay, so you see here, they actually give you, okay, all those are simulated simulation. So here, oh, by the way, I forgot to mention, all those data sets are simulated by our IC design simulator, okay, so that we can know which cell is the true doublets. And same here, we use simulation.
Add a note. So
Yeah.
IC Design 2 was our first ever seminar talk here, given in the second week by Tianyi. So what I meant is that what he used was the simulator called IC Design. That was the previous version of Tianyi's work, and he used that for his simulation. Yeah.
Yeah, because when I do this study, the IC Design 2 is still in development. So yeah. Um, so anyway, you see those red clusters, right? But they're not true clusters. They're just doublets, which should not exist here. And then you want to remove your doublets so that all those fake clusters will gone, right? Will be gone. So and but you are not perfect, so you still have some right points left. But you see, the problem is mostly solved, right? Even if you have some doublets left, but most of them will be removed, and you will not have those fake or superior clusters. So that's our goal. Okay, so here we jump up to our conclusion or comparison. Uh, so basically, this figure shows the x-axis are all those methods, and the y-axis are the doublet rate in our data set. Okay, so we generate a lot of data set, and we simulate doublets, and we let the doublets increase from 1% to 25%. And for each, then we will have 25 data sets, right? And then for each data set, we let each method to identify the correct number of doublets. For example, if we say this one, 20, okay. Oh, I think, oh yeah, so I think I'm wrong, so no. So the data set, all the data set will have 20% doublet rates. Okay, so that's why we label 20 here. But the y-axis are the number of doublets we let the method to identify. Okay, so we let the method identify 0, 1 up to 25% single cells as doublets, and then we remove all those doublets and we do clustering. Try to compare whether the clustering is same as the true clusters. For example, in the first figure, we simulate 4 cell types, right? So the true clusters are 4. And then all those orange grays are the wrong number of clustering result, okay? And all those yellow grays are correct number of clustering result. So we hope a good method can give us most number of yellow grays after it removes doublets, right? So as you can see here, those two methods actually give us most correct number of clustering after applying them. And then we also apply them on 6 cell types, 8 cell types. And basically the comparison is pretty consistent, okay? basically the method on the right side, they generally perform better on the left side. And then this is the effect to remove all those red clusters, right? And we also want to check among all those correctly clustered clusters, how many doublets are still here, right? We also hope a good method not only can remove all those red fake clusters, but also make your correct cluster pure. Okay, so then this is still the box plot of your synced list proportion in your correct clusters. Okay, we hope them higher better. So it's just like the proportion of those gray points in each cluster. Okay, it shows you the capacity to remove all those doublets in each correct identified clusters. So still, those doublet finder and this method doublet detection perform better here, but also we found the screwblit outperform others here, even if screwblit is not very good in this comparison, but it's actually good at remove all those doublets in these clusters. Okay, so then the next one, we want to try to see what's the effect of doublet detection on highly variable gene identification. So what is highly variable gene identification? So, you know, in your analysis, you have tens of thousands genes, right? And you actually don't want to use all of them because most of them will not give you a lot of information because most of them are just a 0 or 1, 2. You want to find those genes that they are very different in different cells. Okay, so we call it highly variable genes. Then those very different genes will give you much more information than say one gene with all zeros, right? So we want to identify those genes. And also, if you have doublets, you will introduce those non-exist cells. It will affect this task. So we want to see what's the effect of this doublet detection on highly variable gene identification. So here, we still generate, we use simulation to generate some data set with 40% doublet rates. And then we first use this SURAT package to do this highly variable gene identification. So you will see here, we first identify those genes on contaminated data, which means this black one is the 40% doublet rates. And also we identify them on the clean data, right? So then of course, the result on those two should be very different, right? Because the contaminated data will include doublets. And then we apply each of our methods on those contaminated data with 40% doublet rates and remove 40% identified doublets. Okay, then after remove them for each method, we will have say we call it after removing data set, right? And then we redo the highly variable gene identification, and we compare the highly variable gene from the after removal data set and compare them with the clean data set. If a method is good, then it should give you a very similar result between these two, right? Because after applying, removing, you should be close to the clean one. So here, the first figure just use the Jerkat index to show the similarity. Basically, you want your method to give you a high Jacob's index to give you more similarity comparing with clean data. Okay, and here the result is that only these two methods doublet finder and scrublet can actually improve the similarity compared with contaminated data. So other methods actually didn't improve this similarity. Okay. And the second figure basically show the same story, but here the value is just the difference comparing with the black one, the contaminated data. So contaminated data, yes. So basically, the message is only two methods improve this task. And then next one, we want to check the gene. Yeah.
Time to pause here a little bit and let the students ask if they have any questions so far. I realize that there might be a lot of new concepts that have been introduced. So if you have a question, probably it's a good time to first sort them out before we move on. I can say the concepts include the doublets, right? That's the fundamental biological concept he's talking about here. And then he introduced 9 methods, right? 9 computational methods, each with a different name. And then we use different criteria to evaluate and compare those 9 methods. So I think starting from here, the important concept will include the highly variable genes, the differential expression, those kind of things. Each concept will be one aspect of evaluation or one criterion for evaluation. So that's why I think if you don't really understand what is going on here, don't be shy. Just unmute yourself and ask. So one thing, yeah, go ahead, please.
So the reason that you need to have this doublet detection for these highly variable genes in specific is that so you can have the more accurate evaluation methods later on, correct? So that particularly in these genes which have a lot of variability, it makes it even more specific for analysis.
Yes, yeah, yes, that's right. Because you have, say, 20,000 genes, right? But you actually don't need all of them because most of them, say 90% of them will be not that useful because they just don't have any difference in different cells. So if they are,
I can add one more thing to clarify. Or we think because those genes variation is not big enough, we do not trust those variation. We think those variation might be to technical noise as opposed to biological variation. So that's why the highly variable gene selection is a necessary step in single cell data analysis. After you select those highly variable genes, any further analysis would just be based on those small number of genes, like hundreds of genes. Yeah. Yep. Okay, so Miles, I think we can continue. Thank you.
Yeah. Okay, so yeah, please let me know if you have any question because I don't know. I'm not very sure what's our say previous experience on those concepts. So yeah. Okay, so basically what we are doing here is just focus on different very common analysis and try to see before and after removing those doublets, what's the difference on those analyses, right? You hope your method can improve all those analyses. So here we move to another one, which is gene analysis, which means differentially expressed gene analysis. So basically what you want is, say you do some experiment, right? You have treatment and you have control, and then you collect the data from these two conditions. Say for each gene, you want to see what's the difference between these two conditions, right? You apply some drugs, right? Then you find gene 1 significantly different from you don't apply that drug. Then you say, okay, this drug have significant impact on gene 1. Then we want to find all those all those genes that give you significant difference under two conditions. So we call them as DE gene. So of course, if you have doublets, your DE gene will be different as your clean data. So we want to see how each method can improve the gene after applying them to remove doublets. So the figure just showed the first one. We use 3 commonly used DE say software to identify DE genes. So DE62, MAST, and Wilcox. Still, because we want to compare the true DE gene and the identified gene, DE gene, so we still use precision, recall, and true negative rate. Okay, precision just give you among your identified DE genes, what's the proportion of them are correct, and recall is among all the DE genes, how many of them you can identify. The true negative rate give you among the non-DE genes, how many of them you can identify. Okay, so still the higher the better, and they are between 0 and 1. So still the black one is the negative control. Those are the result on your contaminated data, which is still we simulate 40% doublet rates, which is this black bar. Okay, and then this say the last one is like brown, light brown. This is the clean data result. So your D analysis on data sets without any doublets. This is we call it the positive control. So of course, all these 3 metrics, you will see the positive control will be higher than negative control, right? Because there's no doublets, so you don't have any problem. Then we apply each method to remove 40% cells they think should be doublets, and then we redo this D analysis to get precision, recall, and a true negative rate, and try to see how we can improve over those negative control, right? Over those black one, and how close we can get to the positive control. Okay, so still the higher the better. You can see all the difference here, but there's one interesting thing is among all those comparisons, you will see the recall actually has the doublets has the most significant impact on recall because they are relatively low, right? You have a 0.6 to 0.7, but for other method, excuse me, for other method, they already very close to 1. So basically, we find the impact of doublets is mainly on recall. And still, we find doublet funder, this pink one, and the red one, sometimes the doublet cells, and the screwblit, they are better than other methods. So basically, you can clearly see which one is better than others. Then the last downstream analysis we want to check is the cell trajectory inference. Okay, so this one is similar to clustering, but you can check you can think this trajectory inference as a continuous version of cell clustering. Cell clustering just give you discrete clusters, right? But here, because we are dealing with cells in differentiation, so there's a very clear trajectory. Some cells are in this stage, and some cells in that stage, and the time is continuous. So if you project them into low dimension, you can see this clear pattern. Okay, there's a lot of methods that you can construct all those trajectories, but those solid lines are your constructed trajectory, and those red points are doublets. And as you can see here, the first one is clean data, no doublets. So we have these 2 branches as our trajectories. But here, the second one, you have some doublets. And then the trajectory construction method will think, okay, we have a lot of points in the middle, so we should construct a 3rd branch here, right, between these 2. But actually, this 3rd one is wrong. We actually don't have all those right points here. So you think a better method should remove all those right points in the middle, and so that the construction can remove. They won't believe there is a third branch in the middle. Okay, then all the figures after this one, each one is for one method. So you can see there's only one method that failed to remove doublets so that the construction method can remove this fake branch, right? But for other methods, actually, they can get that point that there's no fake branch, superior branch. However, even if the results are similar, you can see the actually removal is different. Some methods still have something in the middle, but the number is not that large, right? And some methods actually clean it very well. So we also simulate a sequential trajectory to see whether how the method perform, right? So basically, you have three sequential trajectories, and you have some doublets. And after you remove them, you hope that you can remove all those superior trajectories and still same result, a similar result. There's one method failed to remove the trajectories, the fixed trajectories, and the other method give you still give you similar result. But if you compare them, actually we find these two methods actually give the most similar trajectory as the clean data. Okay, and these two methods, these two figures give you the final perspective of trajectory inference, which is how to identify the temporally expressed gene, because before we check the DE gene, right? So under two conditions, which gene is significant different. Then here is the continuous case. With the time change, which gene will significantly change as time change? Okay, so if that happens, we call that gene is a temporally significantly a different gene, different expressed gene. So still, doublets can bias your finding here, and we use two methods to construct all those temporary expressor gene and to compare their precision, recall, and the true negative rate. So in this case, the difference among different methods are pretty similar. Okay, so they all can improve over the negative control, which is the black bar. Black bar is the contaminated data. Okay, so then we are all done on downstream analysis. And the next one, we want to compare some computational perspective of all those methods, which is distribute, computing, speed, scalability, stability, and usability. So the distributed computing is motivated by the current development of big data, because especially in single cell RNA sequencing, usually sometimes people can generate millions of cells, and you will have a sample size as millions, and the dimension, the p, the variable, will be tens of thousands. So that's a huge data. So then you will have issue to deal with this very huge data, right? So if you want to use your computer, so usually you don't have that computational power to deal with that huge data. So then you have to split the data into smaller pieces, either do them one by one or put them into cloud to do parallel computing, right? Then we want to check under these settings how different methods perform. The idea is, ideally, you can do them at once, right? The big data. Then you can generate for each cell, so say millions of cells. For each cell, you will have one doublet score, right? The higher the cell will more likely to be doublets. Then you will have a vector with length being as millions. That's the normal result. But assume you have to use distributed computing. Say you have to use 10 different computers to calculate this huge data, right? So each computer will deal with, say, 10% of your data set. So it will give you 10% doublet scores, right? Then you will have 10 of those 10%, and then you concatenate all of those vectors. You still have medians, the length of vector with length as medians. But this vector is different from the original computing because you just calculate smaller data, right? So we want to see what you lost in this process, right? So then we do some comparison. The x-axis, this is calculated on these two data sets. So x-axis is the how many batches you split your data. Okay, before we have so say we split them into 2, 4, 6, 8, and 10, and 1 just means you use centralized computing. You do it once. Then the y-axis is the performance matrix. Here we still use AUPRC, and we compare. So the first point is the original computing result, and you see with having more and more batches, which means you split the data more and more, then the performance will be going down. Okay, so the reason of this phenomena is that when you use distributed computing, you kind of lose those global information because you only use 10% of data. So after you combine them back and you check the performance, you will lose those information. So the performance will go down. And so basically, the second figure is based on another matrix, which give us the similar result. But we actually found there's one method which the going down trend is not that significant, which is CXDS, and sometimes it's even going up. So we found actually there's one method which is pretty consistent under different batches. So then next one, we basically compare the computational efficiency of different methods. The first figure just show the distribution of their computing time on 16 real data set. Okay, and also on the y-axis we take log because some methods are actually very, very slow, so time is very long. So you will see still this CXDS is the fastest method. And here we plot the relationship between running time on your real data and your performance metrics, AUPRC. And you see, generally speaking, if you want to get better result, your method tend to be slower. Okay, and then this one show the relation between your sample size and your running time. Okay, so we see most method will have linear increasing when you increase your data set, right? So there's a straight line going up, but the slope is different. There's one method which give you irregular relation. So this one basically is based on neural network, so which is very special. So you have this very irregular relation. And the last one is the stability of your method. So we designed this because different method, because there's always randomness there. So different method, if you run different times on the same data, you probably get different result. So here we just do a resampling, 90% resampling from your original data set, do it 20 times and run the method to get a distribution of your AUPRC and plot them here. You see this pink one is doubly funded with, this is a relative better performance, but this one also have relative high variance. Okay, and there's a method in the middle, and actually we find the interesting phenomenon, which is the red one is the worst method in our previous comparison, but this one is most stable. Okay, so it's consistently perform poor. Okay, so that's the we found here. Okay, so the final comparison is the usability. So basically, we compare the software quality, the convenience to run the method, and the publication and the documentation. So basically, this one just summarize all those methods, and to emphasize, we should take care of how the software implemented, how the user feel about how to use the method. And also we give some score here to quantify this comparison. Okay, so then we almost finished. So we will move on to the summary and discussion. So this figure I made to summarize all those methods. Basically, those spider figure just show all the dimension we have compared, and all those areas just show you the performance. Okay, so and the blue one are actually the accuracy and the downstream analysis perspective, and the black one are the computational perspective. Okay, from here you will see the Doublet funder actually doing very well on accuracy perspective, and but this method is kind of slow. But for this CXDS, it's very good at speed and efficiency, but the performance is just in the middle. Okay, so by this method, you can clearly see how the different methods compare. Okay, so then we have some open questions left to let users and the new method developers to consider. So the first one is how to estimate the exact number of doublet rate in any data set. Okay, because anyway, in your real analysis, you don't know how many doublets you have. So all the methods can give you doublet score, which means how possible each cell is like a doublet, right? But it didn't tell you how many doublet you have. There are some proposals that for three methods, they give you very rough guidance how to decide which how many W you have, but there's no golden standard. So there's one first open question to ask. The second one is how to distinguish homotypic doublets from singlets. So homotypic means the doublet is actually coming from two very similar cells or two cells from same cell types, right? If that happens, that doublet is actually very like singlets, right? It's like you combine twins, then the new person is similar to that any one of those twins, right? However, if you combine, say, two persons from two different countries, that they're very easy to identify. So the question is how to identify those homotypic doublets from singlets. Then the next one is how to better generate artificial doublets, because we see almost all methods, first, it will give you a artificial doublets, randomly pick up two cells from your data set and simulate those doublets and try to see among your existing data set, how many of them are close to those simulated doublets, okay? However, if you combine two single cells, then on average, the new cell will be doubled, right? The gene expression will be doubled. However, this table, we calculate from the 16 real data set, we find that actually the gene expression is not doubled in those two doublets. It's only 1.5 as the singlets, right? So which means if you just randomly pick them up, add them together, you are not getting a very accurate artificial doublets. So people may think about, we should consider all those numbers, right, to make your artificial doublets more similar to real doublets labeled by experiments. Next one is how to ensemble different methods. So as we said before, people believe that if you have two relative better methods, if you can combine them together, then you probably will get a very good method which outperform either one of them. That's people believe. So, and which method you should pick, people usually think, okay, first, those two methods should be relative good. And the second one, those two methods should not be exactly same. So here, to give a guidance, which method we may choose to combine, we compare the similarity of all those methods. So this matrix just show the correlation between two methods in terms of their doublet score. So if they have, say, high, and also say for this one, for doublet detection and doublet finder, you have 0.7 correlation in terms of their doublet score on real data set. So these two methods are very similar, and also these two methods are perform relative good well. So you may consider combine these two. And also, the second matrix is the intersection between your between two methods in terms of their identified doublets. So before we combine, we measure the similarity between doublet score. And the second one, we compare the similarity between identified doublets. Okay, so still, you see the doublet detection and doublet finder have most overlaps. They have the most overlapped identified doublets. So I think the last one is the effect of hyperparameter tuning on your method. So hyperparameter is the parameter to control your model setting. For example, in your k nearest neighbor method, you see, okay, within some distance for each cell, how many of them are artificial doublets, right? And use that one as your doublet score. Then how you can define this distance? So this distance is your one example of hyperparameter, right? You can define them as say 10 or 20. Then it will give you different doublet rates within this distance, right? If you include more, then you probably get different result. So this is a hyperparameter you can tune. So we actually found these three methods are actually very similar. They are all using KNN-based method, but their performance is very, very different. Doublet finder are almost consistently outperform other two. So we suspect that maybe that's due to the hyperparameter tuning, right? The different number of doublets you simulate and the different distance you set up to calculate the proportion of doublets. Then we do one experiment, which is we substitute the hyperparameter of screwblit by those decided by doublet finder, right? So the pink one is the doublet finder, and the blue one is screwblit, and the yellow one is the screwblit with the hyperparameter as doublet finder. And you can see here on some data set, using different hyperparameter actually improve the performance of screwblit, right? Because the yellow one is larger than the blue one. But on some data set, there's no significant difference. And even on some data set, it gives you worse result, which shows that hyperparameter tuning is very important in some cases, but it did not decide all the it not give you all the reasons that differentiate between different methods. So that's the last message we wrote down in the paper to let future developer to discuss, to think about. Yeah, I think that's all for this study. So I hope it's clear. So please feel free to ask any questions or comments if you have. Thank you.
Is there any like future developer, like specifically that you have in mind, like to use this information?
To be honest, I don't have any very, I would say, breakthrough idea to develop new method. However, I actually have one idea that I actually want to automate all those benchmark, which means we can make a package that incorporates all those benchmark studies so that if you have new method, you can just run it, and it method will go through all those process and compare with current method. So then people will know, okay, this new method is either good or not good. So for me, I think that's more interesting direction. Because current study is just a hard code, right? You read the code, you compare, and you write down a paper. So, and that's done. If you have new method, you have to manually go through this process. But I hope there's a tool that you can automatically just run the method, give the method to the package, and the package will output all those comparison results. I think that's more interesting for me.
And actually, Miles, I think probably it's a good idea for you to share the screen of our paper of the discussion section, because over there we did mention some future directions. So students might be interested in reading what we wrote. The main text, the discussion section.
Discussions here. Future direction is
probably just go through the whole discussion section. Just give them some time to read each paragraph. I think that will be clarifying. Yeah, there's no need to go full screen. I think this is fine.
Oh. Okay. Do you want me to quit the full screen now, or?
No, you are not in full screen anyway.
Okay.
You don't want to be in full screen, because when you're in full screen, then the height will dominate the width, and then it will become very narrow.
Okay.
How
about now?
I think it's good. It's just like,
yeah. Okay. Okay, I think this paragraph just summarizes the result and show the impact of our work.
You can just press the down button so to make it flow slowly. Yeah.
Oh, like this?
Yeah.
Okay.
Okay, we can pause here for a few seconds. Or maybe you can read it out. Anyone can read it out. That can keep people on the same page. I don't know which is better.
I will read. I can read. So with the rapid development of scRNA-seq technologies, a skyrocketing number of computation methods have been developed for various scRNA-seq data analysis. For example, since 2018, more than 45 imputation methods have already been developed to recover missing gene expression. commonly referred as dropouts in scRNA-seq data. Such richness of computational methods is a double-sided blade. On the one hand, scRNA-seq researchers have more blocks to build analysis pipelines that accommodate their scientific investigation needs. On the other hand, it becomes increasingly difficult for researchers to choose the method from dozens of methods developed for the same purpose that best fits each step of their pipeline. Unlike in experimental sciences, where new technologies often replace old ones, there are usually no clear-cut or universal choices of computational methods. An appropriate choice of computational methods is case by case, depending on data characteristics and scientific questions at hand. Inappropriate method choices would, to varying extents, bias data analysis, such as by introducing artificial non-biological signals and ultimately lead to false discoveries. To avoid this issue, the scRNA-seq field and the broad biomedical science community yearn for comprehensive benchmark studies that independently and fairly evaluate computational methods. A well-designed benchmark study should offer users objective, accurate, and informative guidance on selecting the appropriate methods for a specific analysis task. To provide the first comprehensive benchmark of computational doublet detection methods in this study, we evaluated 9 existing methods using 16 real and 112 synthetic scRNA-seq datasets from 3 perspectives: overall detection accuracy, impacts on downstream analysis, and computational efficiency. We further categorized our benchmark results in 9 aspects, including 4 related to doublet detection accuracy and 5 associated with software implementation. In summary, Doublet Finder is the best method in terms of accuracy, yet its computational efficiency and stability are not among the best. The CDS method is the opposite. It has the best computational efficiency, excellent stability, but medium accuracy. Our summary is consistent with the aforementioned principle of computational methods that no method is universally the best. So a fair comparison of computational methods should be multifaceted. Although our benchmark study has collected all the available scRNA-seq datasets to date that contain doublet annotations, we know that none of the annotations is utterly accurate due to experimental limitations. For example, the two species mixture datasets, HM12K and HM60K, only label the heterotypic doublets formed by a human cell and a mouse cell. The 6D Maxlit datasets only label the doublets formed by cells of 2 individuals. Many homotypic doublets were unlabeled in all these datasets. As a result, the incompleteness of doublet annotations would have inflated the false negative rates and reduced the precision of computational doublet detection methods in our benchmark. To overcome this limitation, we designed extensive simulations to benchmark computational doublet detection methods in a fair and comprehensive manner. Yet, how to generate accurate doublet annotations by experimental techniques remains an open question to experimental scientists. Regarding the future development and benchmark of computational doublet detection methods, here we list the 5 open questions we deem important for computational scientists. First, how to estimate the unknown doublet rate in a scRNA-seq dataset. Some methods provide heuristic guidance to estimate the doublet rates or select the threshold on the doublet scores. For example, Doublet Finder suggests using the rates of heterotypic doublets and the Poisson doublet formation as the respective lower and upper bounds of the expected doublet rate. Scrupulous recommends setting the doublet score threshold in the middle of the two modes, which it expects to appear in the doublet score distribution. Solo sets the doublet score threshold to 0.5 by default. However, there lacks consensus or direct estimation of the doublet rate from scRNA-seq data. To address this issue, we suggest estimating the null distribution of doublet scores of singlets as a preceding step. With a reliable null distribution estimate, estimating the doublet rate would then become feasible. Second, how to distinguish homotypic doublets from singlets. Existing computational doublet detection methods cannot well identify the homotypic doublets that have similar transcriptome profiles to those of singlets, likely due to the ways they generate artificial doublets. A possible direction is to extract and incorporate features that can distinguish homotypic doublets from singlets, such as the doublet library size. Third, how to distinguish doublets from doublets contaminated by ambient mRNA. Ambient mRNA molecules are released from lysed cell into the cell suspension. They may enter doublets and contaminate the measured transcriptome profiles of these doublets. Similar to doublets, contaminated doublets by ambient mRNA also confound scRNA-seq data analysis. Existing computational doublet detection methods do not distinguish these two types of non-singlet doublets. Instead, computational methods have been developed separately to detect contaminated doublets. Ideally, the single-cell field desires a computational method that can simultaneously remove all non-singlet droplets, including doublets, contaminated droplets, and empty droplets from scRNA-seq data. Fourth, how to improve doublet detection algorithm regarding the use of artificial doublets. The majority of existing computational methods tackle the doublet detection task as a binary classification problem. To train a classification algorithm, they use original doublets in data and artificial doublets they simulate to represent the singlets and the doublets respectively. However, not all original doublets are singlets, because otherwise we would not need doublet detection. By neglecting difference between original doublets and singlets, existing methods do not supply their classification algorithms with quality training data, and a likely consequence is that their post-training classifier would be biased, and thus miss a substantial number of doublets among original doublets. A possible remedy for this drawback is to filter out the likely doublets from the original doublets, for example, by applying outlier detection methods before simulating artificial doublets and subsequently training a classification algorithm. An alternative remedy is to keep the training data, but train a classification algorithm under the learning with noise labels machine learning framework. Moreover, there are possible improvements to be made in the generation of artificial doublets. Instead of simply adding or averaging the gene expression profiles of two random doublets as done in existing methods, finer adjustments can be made to the mixing of two doublets so as to generate more realistic artificial doublets. Fifth, how to ensemble doublets detection methods. As a multifaceted problem, doublet detection can hardly be solved by one single computational method. This is due to the diversity of scRNA-seq datasets. The success of the method hybrid, an ensemble of two methods, BCDS and CXDS, motivated us to think that ensembling reasonable and complementary methods, a technique widely used in machine learning, may boost the accuracy of doublet detection. Supplementary table S11 and S12 show the pairwise similarity of doublet detection methods in terms of their doublet scores and identified doublets in the 16 real data sets. Seeing that top performing methods exhibited noticeable differences, we expect that there is a room for using the ensemble technique to develop a more accurate doublet detection method. By dissecting existing doublet detection method, we found method performance highly dependent on the values of hyperparameters, also known as tuning parameters, if any. For example, doublet finder, scRNA-seq, and doublet cells all use the k-nearest neighbor algorithm to distinguish doublets from singlets. However, surprisingly, doublet finder outperformed the other two methods in most of our comparisons. A probable reason is that doublet finder optimized several key hyperparameters of the KN algorithm in a reasonable and data-driven way. For example, doublet finder selects the number of nearest neighbor k by maximizing the bimodality of the doublet score distribution. This advantage makes doublet finder adaptable to scRNA-seq datasets with distinct characteristics. In contrast, scRNA-seq and doublet cells each assign a fixed default value to k, restricting their flexibility and generalizability. The choice of hyperparameter values is especially important for methods built upon complex algorithms. For example, BCDS uses the gradient boosting algorithm, a leading classification algorithm that has more hyperparameters than the simple KN algorithm does. However, the additional complexity did not make BCDS outperform doublet finder, probably due to the lack of hyperparameter optimization. This phenomenon emphasizes the importance for bioinformatics tools to optimize hyperparameter values in a scientific, data-driven way. Ideally, doublet removal requires both experimental techniques and computational methods. If permitted, researchers may use an experimental technique and a computational method sequentially. That is, they first use an experimental technique such as multiplexing to filter out obvious doublets. For example, the doublets formed by cells of different samples, and then apply a computational method to further screening for the remaining droplets that are likely doublets. Or they may combine the doublet scores assigned to each droplet by an experimental technique and a computational method, as proposed by the method solo. The second approach requires the experimental technique to have a doublet score system. In summary, computational doublet detection is critical for the quality control of scRNA-seq data analysis. Our study is the first comprehensive benchmark of currently available doublet detection methods under a wide variety of biological and technical settings. Our study provides much needed guidance to researchers in choosing appropriate doublet detection methods for scRNA-seq data analysis. Our results also point out directions for further methodological development and improvement in computational doublet detection, an active area of bioinformatics research. That's over here.
Yeah, very nice. Thank you very much, Miles. So I hope this reading of this discussion paragraph can clarify a lot of questions. And so actually maybe we can share with the class about our preprint, right? So we have the preprint of the paper online. So let me do it. So for those of you who want to check further details, you can go to the link. And we expect that our paper will be published soon, probably by the end of this year or early next year. That's the hope, because it's already accepted. So I hope the remaining process can be efficient. And yeah, so let me do it. So if you want to check it out, I'm basically copying the link here. Yeah, it's in the Zoom chat box. You can check it out to get a PDF version of our paper. Yeah, I agree with Miles that it's very, very important that we need benchmark studies in our data science. The reason is that there are so many computational methods that have been developed almost every day, especially for people majoring in quantitative sciences like computer science, statistics, or data science. So, the tradition is that if you're a PhD student or if you're a faculty of professor, then your research should focus on method development. That's regarded as important and respected. However, respectful. However, if you really think about it, do we really need so many methods at once, right? A lot of them were developed to tackle the same problem. Even though each new paper would definitely claim that their method is better than previous method, but often that aspect can be quite technical or quite special. So if you really apply the new method to your real problem, it may not perform better than old methods. So that's why I think third-party validation or benchmarking like Miles did would be very important for the field. Otherwise, users will become more and more confused about which method to choose.
Yeah, so there's actually some top journals. They actually, as I know, they push some special issue that just focus on benchmark. They just announce you can they just focus on a different method. So the whole issue will be benchmark studies. So which means the expert in this field pay more and more attention on the benchmark study. So I think that would be a very, very prominent or potential direction you can try if you want to do research. And also it can give you a very clear image how this field is developing. So because during this process, you can know all the methods in this field very, very clearly.
Yeah, I think that's a very good point, especially if you're considering your future graduate study or research career, if that's in your mind. So you at least you know that there's also a direction, which is about benchmarking computational method, or people call this meta research, right?
Yeah,
So
yeah.
I think Miles is especially interested in this.
Yeah, meta research means the research on top of research. So you have second layer of research. Your target is research itself. I don't know whether it's accepted by every field, but at least in this field, it's very necessary.
Yeah, it is. Yeah. Okay. Any more questions from our students?
I had a question. So when you're doing a benchmark study, do you have to get permission from the different methods and like their creators in order to compare them, or are you just like allowed to do that?
Okay, so that's very good question. The answer is no, because in this field, everything is open source, and also all the authors, they want their method to be used, so there's no restriction. And also this very objective third-party evaluation. So actually, people expect that there's no interest contradiction, right? So they hope there's people that they don't do this method development, that they can evaluate those studies. So you can just use as long as it's open source. So you can just use them. There's no restriction. You don't have to ask them.
Yep, that's very special about academic work. So as long as your work is published, you are required to release your software, your data, your analysis code to an open source repository, so other people can use them freely. But I think the only exception is probably the data related to privacy. So like those medical research papers, they may not release the patient data, and you have to apply for permission before you can do it. But for those computational work, it's actually required by journals too, that the authors release everything for transparency. Yeah.
Yeah, and also actually we have to upload all our data, all our code to open source say website, so that people can check our benchmark itself. So everything is transparent.
Yeah, so that's why Miles was saying that he hoped he can develop a package to make the benchmarking automatic. Then people can easily do benchmarking later, at least for the special topic, for doublet detection topic, because we anticipate that there will be more methods to be developed. So then our benchmark result will become outdated for sure, right? We cannot include more and more methods, but other people, if they want to, or if the method developers, they want to see whether their new method is really better than existing method, they can also use our package for comparison.
Yeah, and I think you can imagine this benchmark study as it's very common in your daily life, right? So you have new phones coming up, you have your MacBook coming up. So you will see a lot of websites, they are doing those kind of benchmarking, right? Before you buy stuff, you just check those benchmark, right? So which product whether it deserve you to pay the money to buy, to update, right? Sometimes it's not that improved, so you can stop, you can wait for another year. So it's just like those kind of benchmark. But here we just very seriously apply them in this academic setting. So you can imagine it's like your daily life.
Yeah, these are very, very good questions. I think especially for undergraduate students, if you have any big picture questions, you should ask them first. And because I've always think before we go technical, we need to really have the right motivation and the big picture about why we want to do this, right? Otherwise, it's very hard to pursue a graduate degree if you don't have passion for the work you do.
Yeah. Yeah.
Okay, so as an announcement, we are going to have our next seminar talk next week because of the schedule swap I mentioned I think in the first lecture. So we'll have a lecture next week, and then the last one will be on December 11th. So that will be the last week of this quarter. So, okay, let's thank Miles again for his talk. And yeah, so and thank you all for your participation. Yeah, so have a good weekend, everyone.
Yeah, thank you.
Bye.
Bye. Bye.
Thank you.
Thank you.
Bye.
Bye.
Thank you, Miles.
Bye.
Thank you.
Bye.
Bye.
Thank you.
Thank you. Bye.